Hello, and welcome back for the second half of this lecture where we're talking about somatic gene therapy. And so we're going to take a look at kind of a relatively quick history of human gene therapy and kind of where we're at kind of at this point. So like we've talked about before, gene therapy studies have to have a lot of review associated with them, even before getting approval for testing them within human beings within a clinical trial. And so the ethics, kind of ethics review that we need to worry about if we're to do a gene therapy treatment is you're going to have the Institutional Review Board, the IRB. Remember the IRB, when we started talking about human subject research, they're the ones that are going to go through and make sure that the scientists that are doing the study are going to be treating the patients, the human subjects, ethically, that they're looking out for their well-being and ideally maximizing the benefits associated with the study and minimizing the risk. There's also going to be an institutional biosafety committee that goes through and is less concerned about kind of the human subjects, but is looking at the fact that we're actually going through and working with DNA and potentially using it to alter the DNA, the genes of a person. And so these biosafety committees kind of go through and kind of ensure that the right things are being done to ensure the safety of the genes and the gene therapy mechanism that's being used. Now, if we want to do this as a clinical trial, again, it's going to be expensive, and so we're going to have to get approval. We're going to have to get grant funds. We're also going to get FDA approval for a clinical trial. And so what we're going to see is that even though we've got the institutional approval, the approval by the place where the person is working that's doing the study, in order to do it actually on patients, on human subjects, we need to follow the NIH, National Institute of Health Guidelines on Human Gene Manipulation and Therapy. So they've got special rules that we need to keep in mind. We need to make sure we follow. There's that recombinant advisory committee, recombinant DNA advisory committee, the RAC that we talked about a couple lectures ago. They're going to go through and again, review, especially because we're talking about doing this in humans, they're going to review the plan for the medical, ethical, and safety issues associated with this study. Just because you don't want to go in and kind of alter the DNA of a person without really understanding everything that's going to be going on. And then finally, the Food and Drug Administration, the FDA, is going to be involved with kind of reviewing and approving those studies that are desired to be moved forward as a clinical study. Now, if we take a look at gene therapy clinical trials, what we're going to see is that there are going to be a whole range of different diseases that have been attempted to be treated with gene therapy. And again, these are clinical trials as of 2017. And so what we can see is a lot of cancer diseases, primarily blood-borne cancer diseases, are about 65% of the gene therapy. Monogenetic diseases such as cystic fibrosis are going to be another 11%. So that's a huge amount. And then a variety of other types of diseases that are being looked at in the effort of kind of repairing the immune system in a way, or not the immune system, repairing the body's cells in some way that they would be minimizing the advancement of some of these other diseases. Now, the first gene therapy trial, we talked about cystic fibrosis in 1993, but the actual first trial, a very, very small trial, was done looking at an immune system disease. And again, like these diseases, they got really great names that go along with them, lots of scientific words, adenosine deaminase deficiency. This is basically an ADA deficiency. They're missing a protein, like we've talked about before, but basically it interferes with the normal development of the immune system. And so what they would do with this first gene therapy back in 1990 is they would go through, they would collect patients the white blood cells from a patient. And ideally, in order to kind of minimize the risk of rejection, again, like a tissue rejection or organ rejection, we're going to use the normal white blood cells of the patient, and we're going to try to do gene therapy on those cells themselves. And so what we're going to do is we're going to take the blood cells from the patient, put them in a laboratory setting. And so we've got it here, where we're going to mix in the therapeutic gene, what they're describing up here, the replacement gene, hopefully get this into the cells, and then take the cells that are now expressing the corrected version of the DNA and put it back in the body, with the hope that if we do this, we would be able then to replace this damaged protein with a functional protein down here by replacing the DNA, replacing the information for building that protein. Now, clinical research on gene therapy has continued pretty steadily since the early 1990s. So we got 1990 down here with about 2 studies, 91 for 8 studies, 14. And so if we look at these clinical studies worldwide, we're upwards of about 1,600 trials conducted or in process of being conducted over the last 30 years or so. And so, you know, there's lots of work being done and looking at this as a possible mechanism for the treatment of disease. Now, like any new method, there's a lot that we need to learn about it. And because of that, sometimes things don't happen the way that we want. One example of that is that a patient in an early gene therapy clinical trial died in 1999. This was a patient that was going to go through treatment for a liver disease. It had this disease, OTCD, again, corresponding to a really nice scientific name, which is almost impossible to pronounce, but you got ornithine transcarboxylase deficiency. Basically, it's a liver disease. And again, it's basically a protein here that is not working properly. And the idea is that if we correct that protein, the cells will produce this corrected protein that or correct the DNA, produce the correct protein, and not have the liver disease. Now, if we take a look at this patient that died of multiple organ failure, which was thought to have been triggered by the gene therapy vector that they use. The adenovirus, that cold virus that they use, triggered a very severe immune response. And so because of that, there were a lot of concerns. When they studied what was going on kind of leading up to the death of this patient, they discovered a lot of things. The first is that not all of the risks were described accurately or at all, in some cases, to the IRB for approval, as well as to the patients in the informed consent process. And so when they were doing some of the testing ahead of time, they had some monkeys that died of the same type of syndrome that we saw within the patient that died. But they didn't, include that as a list of adverse effects. They also, in the earlier clinical trials, remember phase 1 clinical trials, you use human volunteers to make sure that the process is safe. What they saw was that it wasn't completely safe. There were still some human volunteers that were not sick, that did not have this disease, that had some fairly serious side effects when treatment with this viral vector. And then finally, they discovered that not all of the correct safeguards that were put in place were followed during at least this patient's portion of the study. And so because they weren't following all of the guidelines, it increased the risk for this patient, and the patient died. And so because of that, we saw that relatively quickly, the FDA halted all gene therapy trials in a whole variety of studies. Example I've got here is using retroviral vectors for blood stem cells, basically looking at it as a huge risk. Again, that's another example of some of the early problems with gene therapy. They were looking at treating bubble baby syndrome. This is severe combined immunodeficiency disease, SCID. You often see it listed as that, with the idea that they have an impaired or broken immune system. And so they've got to, and they're described as bubble baby because they got to be isolated from the surrounding environment because they don't have a strong enough immune system to prevent them from getting sick and dying. And so the idea was that, again, this is an example of a single gene defect. We can go through, treat it. So they would take the blood cells, the cells involved with producing blood in a patient, take it from the bone marrow, treat it with retroviral vectors, incorporate the correct version of the DNA, the corrected version of the gene into the patients and put these blood stem cells back into the patient. And hopefully those stem cells then would continue to express the protein, continue to express and produce lots and lots of blood cells throughout the individual's lifetime. But what they saw is that some of the patients developed leukemia-like conditions. And so when you take a look at leukemia, normally what you have, leukemia is a form of blood cancer. It's a form where, in this case, these cells that they tried to manipulate up here ended up going into kind of uncontrolled cell divisions. And again, this is probably because when the DNA was put into the cells, it caused a new problem. It caused kind of dysregulation or disrupted of the control mechanisms of the cell. So they developed into more leukemia-like, more blood cancer-like conditions. Over time, the FDA eased the ban as they put more and more safeguards in place to try to prevent the development of risks, recognizing that when we're doing these studies, it's a balance between the risks and the benefits. You want to do something that has great benefit and little risk. And so what we do with these studies is try to figure out ways in which we can minimize the risks associated with it. As of 2017, which was the last time I saw kind of good, kind of updated information on gene therapy protocols, there are a variety of locations. What we can see is that a whole variety of them that are approved, looking at squamous cell carcinomas, that's a skin cancer, ADA SIDS, that again is immune system, a couple blood cell situations here where we're looking at acute lymphoblastic leukemia and B-cell lymphoma, again, trying to treat it in that way. Retinal dystrophy, retinal dystrophy, basically resulting in kind of blindness, a disruption of the normal function within the retina. And so there are a wide range of studies that are going on and continuing to be analyzed. Now, in 2019, the FDA approved a gene therapy treatment for spinal muscular atrophy. This is a leading genetic cause of infant mortality, with treatment needing to occur under the age of 2 because these patients often die very early on in their development. And so normally what happens is that you would have, this is a nerve cell here, it would go out, make lots of connections out here with the muscle cells, and you would have, good kind of tone within the muscle, which is, going to allow the baby to thrive. With this disease, spinal muscular atrophy, what happens is that the nerve cells here don't make it out and make as much connection with the muscle cells. And so because of that, the muscle cells kind of waste away. And that's why it says atrophy, muscles wasting away. They went through, and in 2019, started treating this with a gene. So you're basically going to try to put in the corrected version of the gene into these spinal cord cells, these neurons within the spinal cord that kind of extend out and make connections with the muscles and control them. This is one, like the approval processes we've talked about before. The FDA fast-tracked it, gave it a priority review, and gave it orphan drug designation because it's very devastating for those patients that have it, but it's a relatively small percent of the population that would have this. And so this isn't a major disease that the pharmaceutical company or whoever it is that's developing this treatment is going to be able to market and put out for general use. This is something that is going to be incredibly beneficial for a small number of patients within the United States and across the world. Now, the most successful human gene therapies so far have been those associated with treatment of the severe immune deficiency diseases like the ADA SCIDs that we talked about, the bubble baby syndrome. Again, this idea is that we're going to go through and we're going to take cells from a patient, take cells from a baby, normally take them out of the bone marrow. You're going to isolate the cells that are going to be involved with producing the immune system later on. We're going to alter those cells by introducing the corrected version of the DNA, grow these cells within the laboratory setting, often test these cells and make sure that they've expressed the right version of the corrected DNA, the corrected gene. And if that's the case, you put them back, inject them back into the bone marrow. And in doing so, hopefully what we've done is replace the DNA in this baby's blood stem cells. The cells are going to divide and produce blood cells throughout the body. And if we're doing this, this could potentially be kind of a single gene therapy. This round of gene therapy, by producing it in these stem cells within the bone marrow, the long bones, kind of the legs, primarily the hip, you're able to potentially treat that person's disease for their entire lifetime. And so that's been the most successful so far. So this finishes up an overview of somatic gene therapy. Recognize that it's somatic gene therapy, emphasizing that we are treating the body cells of a patient. And so we're going through, and even though we may be treating a baby, like in that previous slide and that previous ADA SCIDs, but we're not affecting it in a way that's going to go to future generations. We're going to talk about that in the next lecture. So with somatic or body cell gene therapy, what I want you to do, take a look at a Food and Drug Administration kind of a consumer report, what is gene therapy, a little bit on the NIH Genetics Home Reference Guide on gene therapy, and then some advances, challenges, and perspectives on gene therapy, and a little bit of the updated gene therapy clinical trials up through 2017. It was published in 18. And so that finishes up the lecture portion of today. As always, if you have any questions, feel free to email me. Thank you and have a great day.